Welcome to the Zero Input Agriculture podcast. Today I have Dr. Willkeen, who writes under the Substack blog Bactelium. It's fairly new, but he's got some really amazing stuff. This is the kind of person I've been looking for to bring onto the show for ages. They are I guess you call them a backyard microbiologist who is like adding the fermentation and the microbial elements to this grand project of putting uh, organism manipulation back in the hands of the people. And yeah, as soon as I saw his material, I knew instantly that I had to be friends with him. I had to bring him on the podcast, and I had to get him involved in this whole operation. So I don't know a huge amount about your background. We're still kind of new to each other. So I'll throw over to you. Do you want to tell us a bit more about who you are and how you ended up on this fairly unusual path in life?
Yeah, thank you for that. Honestly, I have to say I was always just really jealous of your name, Zero Input Agriculture, because I really want to uh, take it a step further, so that you don't even need land, you don't need fresh water, and you don't need any nitrogen at all.
Yeah.
And maybe if I'm lucky, maybe I can even get them to scrub a lot of their own minerals and stuff like that from seawater.
Hmm.
Um, as you were saying, I am the president and founder of a nonprofit called Bactelium. Um, and a little bit about my background. I am from the American South. I'm actually from Mississippi, and I always grew up on farms. And so my family moved around a lot all throughout the South, but I've lived on usually cattle farms for most of my life with large gardens. So I've been gardening um, since I was, I think, like 5 or 6 years old. Um, and I was always super interested in genetics, and I was super interested in science, and I was trying to breed my own varieties of corn or zucchini or whatever I could get, because my grandfather and I would actually have competitions of who could grow the biggest anything every year, and it would kind of be picked at random. Um, but then So my family doesn't have much education in the background. So my father made it to the seventh grade, and his father before him made it to the second grade, and that's it. So uh, I took a scholarship opportunity. I competed for a scholarship from the United States Army back in 2009. And then I was a contracted cadet, and that got me a college degree, an officer's commission, and a job in the U.S. Army. But as a part of that scholarship requirement, I ended up getting actually a degree in international relations, which was very helpful for me as um, I spent the entire time I was actually on active duty, pretty much constantly deploying to um, sort of new NATO nations and non-NATO nations on the border with Russia, like pretty much constantly, to include Ukraine in 2015 and 2016, but pretty much all the time. And so my understanding of international relations, like what does a treaty really mean? How is it binding? When is it not binding? And what you should and should not do when on someone else's territory. It was actually very helpful, but it was in no way preparing me for a career outside of the Army. And so the whole time I was in the Army, I was airborne, which means that I used to jump out of airplanes for a living. Makes no sense to jump out of a perfectly good airplane. I've been told, but I did it. And so I actually ended up hurting my back, and I got medically separated from the Army, and I had to decide, okay, so I've had this passion my entire life of, what can I get biology to do in a very practical sense. And I'm very pragmatic about my goals. And though I do have a PhD, I'm usually much more interested in what I can do with knowledge as opposed to finding new knowledge for the sake of new knowledge. But if I need new knowledge, I'll go out and get it and use the scientific method. But so whenever I hurt my back and then I ended up getting medically separated from the army, I had to find a new path in life. And I decided between either the defense industrial complex on one hand, where I was getting job offers, and to like take the path that I was really passionate about and get a degree in biology and become a scientist. And I decided to take the hard turn, and I got rejected from every single school I applied to because, of course, I have none of the prereqs. But the whole time I was in the Army, I was, while on deployment, would pack textbooks with me in cell biology, microbiology, molecular biology. And my job was actually pretty heavy on calculus. And I would study. And so one university actually allowed me to sort of just show up and convince them that like my transcripts might say one thing, but I actually know what I'm talking about. And that was San Diego State University. And they gave me an opportunity where I just had to take the most advanced biochemistry course they had at the school as like a probationary sort of test in the first year, and then I would get full acceptance. And yeah, I passed that class with an A, and then I was settled into the PhD program. And so my background scientifically is really focused on complex communities of bacteria, especially how the bacteriophages and viruses in that community affect the dynamics of the community. And specifically, I studied cystic fibrosis, which is a very complex chronic lung infection. Oftentimes, whenever people have cystic fibrosis, and oftentimes they're going to have extreme levels of antibiotic resistance in their infections. And so I worked on one, understanding the ecology of their infection, and two, how could you try to address that? And so I kind of had a twofold thing where I would make these models to help us understand the ecology and like, why are those microbes there? What are they doing? Why this one and not another one? And then I would also engineer novel phage-like constructs that could go in and very specifically kill certain bacteria. And it's that genetic engineering bug that caught me and made me realize that, hey, actually, there's a lot of untapped potential for really just practical purposes. And so it was near the end of my PhD in 2023, I came up with an idea that actually I was prompted to produce a moonshot idea. Like, if science is going to solve a problem for the world, how would you do it? And so my moonshot idea was essentially that I could use safe to eat fermentation bacteria to break down indigestible plant parts and convert that biomass into useful food products like starch for gluten, for ovalbumin, myoglobin, and casein. And it's essentially a project that looks at a bacteria and it's like, how close can I get this to be a cow? How close can I get this to be a hen? How close can I get this to being a stalk of wheat? And the answer is probably pretty close, because you can just feed them carbon, nitrogen, phosphorus in the form of stuff that you can't eat, and they will convert it. And there's an efficiency loss, but there's an efficiency loss in all things.
Yeah, yeah.
So,
I think
um,
I compared this to teaching. Well, the model in nature is the leafcutter ant. So they're extremely successful ants that convert inedible, like tough, toxin-filled leaves, and they cultivate fungal gardens underground through a whole bunch of behaviors and chemical secretions, and that's their primary food source. So it's a neat
right,
little trick, and humans haven't quite figured out how to do that. We do little bits of fermentation here and there on the side, but it's never been a central part of our agriculture.
right. And I think that that's something that can pretty easily change because the microbes that we like to eat and are safe should be amenable to modification, which allows us to get them to produce things they otherwise wouldn't. Um, and then there's also another step of making them able to access more of the carbon in those inedible plant parts.
Yeah. Yeah.
But what really compelled me to take this strategy was a conversation with my mentor, Dr. John Mokili, who's from the DRC. Um, and he was explaining to my professor who challenged me to make this moonshot project. He was like, "Why not just feed it to a cow?" And he was like, "It has to be something that anyone could have, and cows are just not accessible
Mm.
to a large number of people. And there's a lot of places where you just won't be able to keep a cow alive, where you do need to be able to keep people alive." And then so I designed a construct, um, and that first plasmid is actually named after my wife. It's called Pa Florentina, because she paid for it because she believes in me. And that first one, it didn't work exactly as planned, but that it's not that it didn't work because um, that it didn't produce starch or that it didn't produce gluten or that it didn't produce the sort of extra carbon source degrading enzymes. It was more that um, the genetic construct was not stable in the target host. And so since then, I've kind of been reworking that. And in that process, I got invited for a postdoc to try and see if I could do this not in fermentation bacteria that we like to eat, but in a methanotrope, which is a bacteria that actually fixes carbon, but it fixes carbon from gaseous methane rather than gaseous CO2. Um, and
I'm
so
guessing
I
in that kind
was,
of system, you would take even cruder biomass and have that in one chamber to ferment off the methane, and then pipe the methane into the higher, the more refined organism that can use that methane as a feedstock.
um, that would be ideal,
Mm.
right? In my mind, I imagined like uh, a methanogen, which is like the methane producing bacteria, living in the same chemostat as an acetogen, preferably something that utilizes CO2 as its carbon source, like uh, Cyanococcus, which is like a cyanobacteria that can be engineered to produce lots of acetate.
Hmm.
Um, And then you can convert, right, half of that um, acetate, which is a 2-carbon fatty acid. You can convert that into 1-methane, and you can release CO2 back into the upper half of the um, chemostat where you have your your acetogen, and then you can harvest the methane for your methanotrope. Um, but yeah, what's great about that specific methanotrope, which is um, methylative microbium alkaliphylum 20Z, and that was my postdoc PI, Dr. Kalyuzhnaya's baby. And that one is a high pH and high salt tolerant methanotrope. So, and it's perfectly happy growing in seawater. And so my original idea there was just that, well, if I can get it to produce starch and gluten in seawater, well, there's a whole lot of countries that have vast resources in methane, but are currently burning that methane in order to fix nitrogen and desalinate water and grow wheat and provide for cattle in a desert where you could just produce a lot of those products in fermentation vessels right there on the coast, or even at that like natural gas source, which would at the very least just be much more energetically efficient. And so you would end up with way less methane utilization for the same end product.
Now, I want to mostly focus on your microbiological work, but I know you also breed a few plant crops. So do you want to tell us about, just for all the people, like most of the audience are plant breeders or leaning in that direction, they're more familiar with it. So I just want to kind of ground things because I know you are still breeding plant crops as well too. So do you want to briefly tell me about probably your favorite project that you're working on?
Right. So the favorite project I have going right now with plants is my starchy squash project. And that one happened essentially by serendipity, because I am working on producing starch in microbes. I have a bunch of iodine at home, which is a reagent that you use to test for starch concentration. And I was growing a hybrid patty pan and a non-hybrid patty pan and a bunch of other pepo squash one year. And then the next year I saved seeds and I planted them out, and one of them produced a squash that was just really starchy. And then I tested it with iodine. I was like, that's like as starchy as a potato. And so I turned it into what we called squash mash, and we had it like mashed potatoes, and that was great. That phenotype I have not been able to recover from the pepo side. But I then started looking at all the squash in my garden, and testing them with iodine to see which ones had starch and which ones didn't. And the majority had none. And same with store-bought squash, like there's almost no starch in a lot of squash. But I had a couple of these like tan cheese style moshada squash that came from a seed provider who collects seeds from Native American tribes in the American Southwest. And this guy was just chock full of it, and it was a deep dark orange, and it was delicious. And so I've since used that one and its progeny as my sort of main focus. And that was like a couple years ago. And this past year, I expanded that population, and then I saw how many squash I could produce and how long they would keep. And I just took like 10 or 15 squash that were produced. I left them out in the San Diego winter, which is not that cold, but it is wet, which is usually when we get all of our um, fungal growth, because during the dry season you'll get essentially nothing. And I had one squash that was 24 pounds of starchy, delicious meat, and now that one was the parent of this next generation. And the way I'm going about selecting for the squash to be adapted to my location, because I have just a suburban backyard, I don't have enough space to really do large trials, except that I'm doing guerrilla gardening, and I literally just walk around my neighborhood, and I find places that are not irrigated and are mostly left alone, and I just spread out thousands of seeds. In the winter during the rainy season. And so this year I managed to recover 6 seedlings that managed to germinate in the cold wet rather than the warm wet. The sort of Mediterranean climate that we have here, which squash really don't like to grow, or the squash varieties that you will like to eat will mostly like to grow in the summer. Here we have coyote melon, and of course we have the manroot vine, which are both cucurbita and grow just fine in our winter. They actually grow pretty much just in winter. But these guys hate to germinate in cold wet. And so I got 6 who survived the gophers and the insects and the absolute neglect. And then I left them for about a month of the dry season to see if they could tolerate a bit of dry. And then I rescued them back to my garden, and then I'll do that again. But I'm only keeping the seeds from whichever ones have a lot of starch. But hypothetically, I could get, one squash to produce at least 100 pounds of good starch that also happens to have a lot of beta carotene and other like good nutrients in it that stores for like 6 to 8 months.
That is amazing. And was there any hybridization at the root of that? Is it an interspecies hybrid, or you're not entirely sure about where that first new trait emerged?
I'm uncertain. So I do pretty sophisticated lab stuff for the bacteria, but when it comes to the plants, I'm mostly I'm not validating the
lineage.
anything.
Yeah.
The lineage at all. Right?
Yeah.
I'm not going back and sequencing anything.
Phenotypic selection for the win. One thing I really wanted to ask you about is the practicalities of these starch assays, because I want to apply this to my canna crop. I'm starting to generate all of this canna diversity, and I'm wary if I just select the biggest, nicest looking tubers, that's not actually what I want ultimately. I want starch. That's what the crop is for.
Right.
So do you have any guidelines on how I could approach that? Like, did you do like extractions, soluble extractions, or microscope sections? Were you like visually inspecting starch grains?
That mostly wasn't worth my time. So in my garage, I have a microscope and I have a decent amount of equipment, but I noticed that actually the ones that have the most starch are whenever you slice them open and you drop a bit of iodine on there, they turn black, just so deep blue purple that they are black. And so I usually compare them to things like sweet potatoes or any other like starchy vegetable, and then I just visually compare them. Because that's a lot of time, because starch extraction I mean, your amylopectin, the branched glucose polymer, is really pretty much insoluble.
Mm.
Um, unless you go through like a painstaking process of like heating it under high pH for like 24 hours, and then you can get some of it to come into solution.
Yeah.
Um, if you just use really, really hot water, you can get the amylose into solution for a little while, but it might crash out if you look at it funny.
Hmm.
Um, so it wasn't really worth it. I did try just boiling it and then testing um,
The liquid and
the
the whole
the
sample. Yeah.
liquid,
Yeah.
right? And then you'll get an idea of how much, right, amylose was able to be solubilized, which will give you an idea of how much starch there was in the original product in case you are past the point of saturation,
Yep.
right? Whenever you just start adding drops there. But if you're past the point of saturation, then you should just stop working and go do something else with your life because you won.
And I think you've given me a mental model of how I can do this. Now, I'm always telling people like you have a range of like highly sophisticated sensing instruments in your head that are like they
Mhm.
rival anything that you can find in like the most amazing lab. The only problem is the lab is really good at churning out numbers that you can put into papers that other people on the other side of the world can compare. Your subjective
Right.
impressions aren't as good at doing that, but you only need enough information to decide between plant A and plant B, which one is actually worth continuing breeding. It doesn't need to be an absolute number. So I think the way I can probably do this with my canotubers is have an iodine solution that I know what the concentration is, and I'll have a color bar. You know how you see those little like they're a little printout with different shades of a color, and I can use
Mm-hmm.
that to put the iodine on a cut cannutuber, look at which color it lines up with, and then that gives me a kind of semi-quantitative way of making informed decisions about which plant is doing what in terms of starch content.
Uh, that's true. But I think you're a biochemist,
Mm.
and you're not going to need that color bar after like one or two
Yeah,
times because your calibrated eyeball will be more than enough.
yeah, yeah,
Because again, like there aren't that many steps between, some starch, a bit more starch, and then saturation. It's really easy to saturate, and it's really hard to dilute because it's not soluble.
mm,
Um, so, like once you're at the saturation point, there is no more um, sort of depth that you can reach
yeah, and then you can focus on other
without doing
traits.
an extraction.
Yeah, then
Yeah.
you can focus on other traits. Okay, that's really good. I'm so glad we talked about that one. And yeah, the emphasis on staple crops that grow, that like want to grow in the conditions you have, that you don't have to do
Mhm.
all of this bending over backwards to keep them alive and producing. It's such a different mindset. You mentioned too with the guerrilla gardening of scattering seedlings around the neighborhood. Do you suspect I've got a theory that when seeds germinate in situ, they go through a period of plasticity in terms of their microbiome, and that different soils can offer different microbes into that relationship when it's forming. And did you bring those seedlings back into your garden? And did you suspect that there might have been a microbial transformation through that exposure?
Um, there could be, though I don't know what it would have been.
Mm.
I know that when it comes to my specific garden, um, I've already done a bit of microbial enhancement, which makes me think that, it would be hard for me to notice. And that's
Yeah.
because my um, uh, postdoc professor, Dr. Kalyuzhnaya, has a separate project at her lab that where I was actually the guinea pig, um, where they isolated methanotropic bacteria from the deserts of Anza Borrego. Um, and whenever you add it to plants, It makes them much more drought tolerant. It makes them much more heat resistant, and it makes them grow faster and yield better um, for no explicable reason, but it
Yeah.
is reproducible. And so we were working on trying to find a mechanism, and while at the same time trying to um, push it out and find commercial uses for it because it worked really well. We just don't know why, um, which is fine. But it means that you know when I was given the opportunity, when the experiment was over, I just saturated my garden with that stuff because it's great. It's great.
It brings up a really interesting question. Like, if this microbe enhances the function of that local ecosystem, why isn't it always already there? And like, why is it only in this really weird, tiny pocket of the desert that you happen to sample? Do you have any theories about that kind of microbiological ecology? Like, I mean, how easy would it be to wipe that new microbe out of your garden by, doing something foolish like plowing it up or, applying salt or something?
I don't know.
Yeah.
I wouldn't have a clue as to like a definitive answer on those things. I
Yeah.
have my
Yeah.
own hypotheses, and so um, I think that it's coating the root, and that we have evidence of, but whether or not it is actually living in between cells or maybe forming like a small cytosolic bridge, and like getting some carbon from the plant itself in order to perform some services for the plant that it can't do itself. We have no idea.
Yeah.
I would say if you don't pull up the plant, then I have not seen any loss of functionality.
Mm.
And so the plants that were treated and stayed in the ground ever since then have continued to
To
grow
prosper.
exceptionally well
Yeah.
under terrible conditions. Yeah, we do not know why it's doing that, but we have a bit of evidence that it is not consuming methane anymore
Mm.
when it is associated with the plant's root, because I tinted my garden and measured methane concentrations um, over like a 3 day period, and it was not pulling down any methane
Yeah.
from the atmosphere that we could measure or that was in any way statistically different from the control plots.
I just realized a point about the dark matter of microbiology. So something like 90+% of microbial species can't be cultured. So they're living out there in the environment. But if you try and put them in a petri dish, they're just like, no, I'd rather die than like live there. And like there's a whole field of people trying to come up with really crazy conditions to convince particular microbes to grow in the lab. Um, and layer on top of that that we only, as a species, discovered the existence of microbes a couple of hundred years ago. And project that onto, like, imagine if we'd only discovered plants, like they'd been all around us and we only just noticed them 200 years ago. And that most of them we couldn't even grow, like, we didn't even know what they were. And I don't know, it just makes you realize there's this like iceberg of like, biological dark matter behind evolution and ecology and like everything that is still relatively new to us and unexplored. I find that really exciting.
So the the way we would talk about that, or what I cautioned some people was that it's kind of like when talking to scientists about the 90+% microbes that are currently not in culture. But we can do metagenomes and we can see their genome and we can understand things about them in silico, meaning we can make computer models of their genomes. And that's one
Mm,
of the things I
mm.
specialized in. And so we can get an idea of what they're doing for the ecosystem. So we have pretty good ideas about them, but my question is like, a lot of the researchers I would know or would talk to would want to try and bring them into culture, and they might spend their entire life coming up with the exact media and the exact protocol that will bring that microbe into culture. And the question is kind of like a dog chasing a semi truck. What are you going to do when you catch it?
Hmm.
Right? And so why? Because um, my lab and myself, we focus much more on uh, holomes. So bringing the entire community and making um, a new home for the entire community in vitro. And so that meant making very complex culture systems with very, very rich media that very closely approximates the source where that community grows naturally. And sometimes you have to use that genetic information and those in silico models in order to build a really good media. Um, but you can just study the whole community together because every community usually has a few things that grow really well um, in vitro. And if you only, take a petri dish approach and you have like 5 different media, you're going to miss out on a lot of them. And they may be doing something important for the community, which would explain some emergent phenomena that you observed in the community, but you do not observe from the individual cultural parts. And so we would just graft the entire community into um, a new culture system.
Yeah.
And we
Yep.
would tweak the culture system until we got them all according to the metagenome. And then why do you need to bring them into individual culture?
Yeah.
Because you can
Yeah.
do your experiments right there.
I mean, that whole microbial community is almost like a super organism where all the different species are particular organs that specialize in certain transformations and functions, and they all kind of talk to each other and interact to make one greater whole. So, I mean, it's almost like
Right,
it's easier to keep a person alive than to culture liver cells in a dish.
right.
Mm,
And so uh, you can think of it as like very complex software, so to
mm,
speak. And if you have, um, uh, or you can think of it a bacteria as a bag of enzymes, or you can take that bag of enzymes and you can turn them into a data matrix, and you can observe dependencies, right?
mm.
And you're always going to have microbes, like take E. coli. E. coli grows like a champ just about anywhere on just about anything, because, well, you can grow it on salts, right? You can give it ammonium chloride and it can make all its own amino acids. You can give it sodium phosphate and anything that needs phosphate, like in the DNA or phosphorylated proteins or phospholipids, it'll make them. And then you just have to give it a carbon source and you have to give it a source of sulfur, some magnesium, your trace elements and stuff like that. But it can handle all of that stuff on its own. So it has essentially no major dependencies. But if you try and run a piece of code that has a ton of dependencies,
Mm.
so it requires some complex form of nitrogen, or it requires some complex form of phosphorus, or it has just one very specialized carbon source that it will eat, and it only likes it when it comes from microbe, X, which you can't even culture, right? And so that code will never run unless you have all the dependencies.
Yeah, yeah,
Uh, and so it's the same. Those microbes depend on stuff that you don't know what it is.
yeah.
And it's likely that one of the other microbes in the community is secreting that and providing it to um, your uh, your community, which allows it to persist and grow
It's a
in
it's a
some
little bit it's a
low
little bit like
relative abundance.
it's a little bit like a rare orchid that needs a specific solitary bee that turns up at just the right time to pollinate it, compared to a dandelion that just self-pollinates in the crack in the pavement. It's quite capable of taking care of itself.
Right.
Yeah.
Yeah. So, um, yeah, if you have a compelling reason to bring something into culture,
Mm.
then you can do it. But I would say that it's probably not worth it because you can just take the whole community and then you can do your experiments on the whole community. And if you're interested in one gene or something like that, because you've noticed that some enzyme has incredible powers or something like that, it can do some new thing that you would like to industrialize, um, then you can just take the DNA and put it in E. coli and study it in E. coli because that'll save you a ton of time and money.
And the funny thing is that microbial communities do this themselves all the time, that they'll take a gene that does something important and transplant it into a different strain in a different community, like in a different ecosystem at every opportunity. So humans doing that in
Right,
parallel isn't really something new or unnatural. It's just a different kind of relationship between a macroorganism and a microorganism.
right. Which actually, so when I was reading your book and I saw the section on Frankenstein's toolbox,
Mm.
it really made me think of um, a sort of a witch doctor 500 years from now who's maintaining an inherited library of Bacillus subtilis spores, right? Bacillus, have you ever worked with Bacillus subtilis before?
I've mostly worked with E. coli, boring old E. coli in the lab, but I've seen it around.
Okay.
Mm.
Right. So Bacillus subtilis is essentially the gram-positive version of E. coli. It's the best understood um, gram-positive, but it forms spores. And those spores, we don't even know how long they can survive um, if you just bury them in a jar in the ground or something like that. But it's
Mm.
for sure decades, if not hundreds of years, um, and maybe even longer. But so you could actually build a library of useful products that can be produced in Bacillus subtilis because Bacillus subtilis um, is what we call naturally competent. So Bacillus subtilis at the end of its growth phase, so if you have logarithmic growth and then right there at the end before it goes into stationary phase and it just has exhausted all the nutrients in its media, it will naturally just look for pieces of DNA and it will pull them in through its cell wall and through its membrane. And then it will look over its genome and find a place where it fits and it'll just stick it in there.
Hmm.
And
Hmm.
so it is meant for horizontal gene transfer, but then you can take those genes of interest that you liked, like let's say it produced an antibiotic that you like, or let's say it produced a flavor that you like, or it produces some useful compound that you like. You can just let it keep going until it becomes a spore. You could literally just bury that spore and you could have your own pharmacopoeia just inherited and maintained over time through Bacillus subtilis, which can grow. It is not picky
Mm. Mm.
on what it can eat.
It's used in
The
some
traditional
food.
fermentation is,
Yeah, it's koji, isn't
yeah,
it? Yeah. Yeah, yeah, you just grow it on
it's
rice
not
and
koji,
salt.
it's
Oh,
natto.
oh, natto. Oh, that's right. Yeah, yeah,
Natto.
yeah.
So
Yep.
it's the slimy.
Stinky
So
soybean.
that's right,
Yeah.
because it produces ammonia. um, so it's not. It's very easy to work with, and I love it because it produces spores. But Bactelium doesn't use Bacillus subtilis. Specifically, I use um, Lactiplantibacillus plantarum, which is the one used to ferment pickles
Mm.
or make sauerkraut or something like that, because it doesn't. It is not suited for the Western palate, let's say, and also it's an aerobe, so it has to have oxygen,
Mm.
whereas L. plantarum doesn't need that, which means that you can go more low tech
Mm.
because you can have a large fermentation vessel and you don't have to stir it or mix it constantly, which if you want to grow Bacillus subtilis or E. coli, you have to either have like a very thin layer so that it has constant access to oxygen, like on a petri dish, or you have to you have to have a bioreactor. You're going to
Yeah.
have to be able to either bubble oxygen through, or you're going to have to stir it.
Mm. Mm. Now, we could go in two directions here. We could start looking at, like, I'm really curious about the idea about how low tech fermentation technology can become, both in like the research and development stage, but and the production stage as well. Or we could talk about, like, in more detail about the kind of things that you're hoping to create with your micro breeding, for want of a better word, projects. It's probably better to focus on like just solidify what are you aiming to achieve with your microbial breeding and maybe what you've already started doing.
Right. Yeah, the two go hand in hand. So I started thinking about how you could do um, fermentation for an individual who is living with essentially nothing in terms of infrastructure, all the way up to how um, a nation state could use um, Bactelium's uh, genetically modified bacteria to supplement or make a crutch for their own industrial agricultural systems. Um, so I have thought about that in depth, and those informed why I selected certain microbes and certain products that they would produce.
Mm.
So to start off, um, Bactelium, again, is a nonprofit. I'm a researcher, I'm a scientist, and I am using DNA, foreign DNA that I select in silico. So I go in a protein database and I look at protein sequences, and then I back translate them into a DNA sequence that will work in bacteria, because bacterial genes are much simpler than eukaryotic genes, but pretty much all the genes I'm trying to produce are eukaryotic. So you got to cut out all of those introns. Um, and specifically, then I am putting those onto an episome, which is like an artificial chromosome. And so it's essentially taking um, a bacteria that has one genome and just one copy of its genome. And then essentially making it have two chromosomes rather than one. And that system is not as good at producing tons and tons of product. Usually you want a very high copy number plasmid, but those high copy number plasmids are not stable over time. And so the artificial chromosome allows me to ensure that when I, release these into the world, you don't have to use antibiotics to maintain the
Hmm.
plasmid.
Yeah.
Um, and it's going to stay there for essentially ever, right? Because it's using the exact same machinery that the bacteria uses to replicate its own genome. So whenever it goes to replicate its own genome, it's going to be forced to replicate my artificial chromosome. And then on that artificial chromosome is where I'm inserting genes that force it to produce starch. And so I've gotten methanotrophs to produce starch, and I've gotten several different gram-positive bacteria to produce starch. Strep thermophilus, the yogurt bacteria, um, lacticase bacillus rhamnosus, that one produced a ton of starch. Um, but those are much easier to transform. But uh, the original plasmid that I made, again, for Florentina, the one named after my wife, that one was not stable. It would stay there for maybe 2 or 3 generations, and like bacterial generations, and then it would just give up, and it would stop producing starch. But in the meantime, it had produced a lot of measurable starch that I could observe. Um, I can get E. coli to consistently produce gluten. So I have inserted both glutenin and gliadin onto that artificial chromosome, which also works in E. coli because essentially everything needs to be able to work in E. coli whenever you're doing molecular biology, because it's kind of like our DNA factory. Um, and then, but I have not been able to transfer that artificial chromosome so far to its target host, L. plantarum. Because the currently that's just a money issue, money and time, because the nonprofit just started on June 29th. Even though the project is much older, I need to gather resources and get enough money to pay for my time or to pay for a lab tech's time and to pay for the equipment, either an electroporation setup or the time for me to work out a protocol where I use conjugation. And that's what I'm currently working on, because it's really easy to get DNA into E. coli. And there's a strain of E. coli that exists for essentially nothing more than producing sex pilus and inserting your DNA of choice into whatever is on the other end of that sex pilus.
Oh, wow.
Um,
Nymphomaniac E.
so
coli.
I know, but I have that strain, and that strain is cheap and easy to work with, and I can do transformations using products that I can buy from the grocery store, namely PEG, which is polyethylene glycol, which is a laxative, and magnesium sulfate heptahydrate, or Epsom salt, which is also a stool softener. So those ingredients in combination are really good at getting DNA into E. coli. And then the step that I've not been able to take so far because of just time and resources is getting that E. coli to transfer the artificial chromosome into L. plantarum, because L. plantarum is hard to work with. In terms of like genetically, it has a very thick cell wall. It's not naturally competent, and it's very difficult to transform. But the reason I chose L. plantarum is because L. plantarum is sort of king of the hill in terms of its own fermentation succession.
Yeah.
Because the fermentation of any vegetables in salt water usually start with Leuconostoc, Mesenteroides, and then you're going to have L. brevis, and then you're going to have L. plantarum, which is actually going to do the majority of the fermentation.
Yeah. Yeah. Once the pH drops below a certain level, it's the only one that can handle the salt and the low oxygen and the acidity.
Right. Because I've been able to transfer this artificial chromosome easily to L. brevis, but I don't want to use L. brevis, at least not right now, because, well, it'll just be replaced by L. plantarum, and then the interesting part of the fermentation is done too soon. And I would have to maintain its purity. And that is not something that's ideal for an individual who's living with no infrastructure. And so I need to make sure that this highly abundant, highly resourceful and resilient microbe, L. plantarum, that is also safe to eat, is the one producing the proteins and the carbon compounds of interest.
For an analogy for people who aren't microbiologists, this is a little bit like how gum tree forests drop all of that flammable wood and then it catches fire periodically, and all of the plants that aren't gum trees like struggle to live in that new kind of ecosystem. It's a self-reinforcing, self-building kind of dynamic that these bacteria produce acid that prevents all other bacteria from hanging around very long. and that's a way of having
Right.
a simple, safe fermentation.
It's kind of like allelopathy,
Yeah.
um, which I'm not scientifically a plant person, but I've been a gardener since forever. And so when I tried to explain that to the microbiologists at the lab, they're like, what is allelopathy? And I was like, cedar trees and how nothing grows under a cedar tree. And they're like, what's a cedar tree? And I was like, well, then, maybe I should come up with another analogy.
Is this a
Um,
bird? That meme.
yeah,
They've been in the lab for too long. Um,
yeah.
oh,
So,
I have a general question. Yeah, I think I'll drop it in now. So you're taking a very detailed approach to this engineering. Like you have specific DNA sequences, specific genes. It's using very high level controlled technologies to aim for a specific end result and then like doing everything you can to get there. What do you think about the alternative possible strategy and possibly technologically leveraged as well of like agnostic microbiology where you're doing like mass selection of multiple strains based on phenotypic traits rather than like gene sequences? Have you seen that work? And like robotics, microrobotics could be used to do that kind of thing on a greater scale, rather than taking hundreds of years of tasting all the different koji cultures until one of them is like, oh, that one tastes much better. And my children didn't die of that weird form of cancer that they all get when they're 40. That must be a
yeah.
better one to go with. Is there a way that we could potentially use intermediate levels of cutting edge technology to do uh, screening based on traits rather than reductionist genetics?
So, having done some screens in my life, um, I hate them. I, and that's mostly because they are a lot of work and I just don't have the time. So
Mm,
genetic screening for me is very difficult to justify when if you are a bit more clever and you think longer and you design things correctly, then you can use the power of selection.
hmm
Um, and selection is for me, like currently, I'm doing a completely agnostic uh, phenotypic selection on uh, Arthrospira platensis, commonly known as spirulina. So if you eat spirulina powder or if you grow spirulina, it's that cyanobacteria. Um, and so I took the spirulina strain that I am interested in and I adapted it to growing in saltwater because it's normally freshwater, alkaline bacteria. And then I've made a ceramic chemostat. So that's very low tech. I literally made the clay myself. I went in the backyard, I got dirt, I levigated that dirt until I selected the clay particles. And then I made a ceramic bowl. We had just a fire in the backyard for kicks and giggles, and it was a fun time. And then I took that ceramic bowl, and now I flow uh, uh, filtered seawater through that system, where only the seawater is making it to the microbes that are living inside of the bowl.
mm.
Um, and so I have some saltwater adapted spirulina that I just inoculated into this bowl. And it's been going for about 6 months now, and I'm starting to get robust um, growth in the absence of nitrogen
Yeah.
because what I'm selecting for is nitrogen fixation because Arthrospira platensis or spirulina does not fix nitrogen. It cannot form a heterocyst. Like the heterocyst is an anaerobic section of one of those spiral filaments that allows for nitrogen fixation because nitrogen fixation only happens in anaerobic environments. And of course, an oxygen producing, bacteria is not going to be able to fix nitrogen unless it has like that set of genes that allows it to wall off a section of the filament for a no oxygen zone.
Mm.
Um, and so ceramic naturally forms somewhere between 0.22 and 0.45 micron filter all by itself, as long as you don't fire it too hot. So a low temperature ceramic is actually a really, really good filter for keeping other bacteria out, but allowing viruses in. And so what I'm doing is I'm allowing a bunch of cyanophages, we call them. Cyanophages are bacteriophages that infect cyanobacteria. Cyanobacteria, again, are the photosynthesizing bacteria. You find them in fresh water, you find them in salt water. In salt water, you're going to find a whole bunch of them that fix nitrogen. Mm. And some of them will carry genes from the host bacteria that they will then transfer to their new host.
In the spirulina.
In the spirulina. And so right now, I hadn't checked it in over a month, and I checked it just before we had this talk. And I've got like a big green biofilm, like gob of spirulina in there. And so I assume that I've caught a nitrogen fixation and heterocyst forming set of genes.
Yep.
Probably not from one virus, but probably from like 5, 6, maybe even 10 separate viral infection events that it took over this 6-month period where there was almost no growth.
And like this is what I think is really powerful. And I'm so glad you have this example, because in natural selection, when organisms are put on the spot, when they have a problem that they need to solve, and they're given an opportunity to solve it, they're not just immediately outcompeted or killed by other things. They're kind of kept on the edge of survival, and they have an incentive to fix that problem and an opportunity. And then all of that genetic information is flowing from all the microbes and other organisms in their environment. It's just a matter of time before they find the right combination of factors cobbled together, and yeah, some among them will solve that problem and suddenly overgrow all of their struggling competitors. And the potential for humans to set up those conditions artificially, but then allow it's almost like what's the best way of putting this? Like we're creating the microcosm that allows a particular kind of evolution to happen. And that, I think, is a really unique thing that humans can do that can change the whole nature of evolution.
Yes. And I think, again, but it is important to design it correctly, because like an example, like the difference between screening and selection, right? The ocean water, which is the only source of nutrients for this spirulina that's growing in my ceramic bowl, is very nitrogen depleted. Also because of the ceramic. Ceramic itself will absorb nitrates out of the solution, especially when the solution is high pH and the seawater is at like 8.2. So it's very, very nitrogen limited. But if I were trying to screen for this, and I were growing all of those microbes in a nitrogen supplemented permissible condition, and then I were trying to just add a bunch of these viruses and then trying to pick out which one is the nitrogen fixer, I may not be able to do that in my lifetime. But because of the way I designed it, all I had to do was wait, and then it was there. And I literally had to do nothing. And then the only way to know that it works is that, well, it started growing and there's no nitrogen. So it's got to be getting the nitrogen from the air. And I did something similar during my PhD, actually, when I was looking for. So during my PhD, I did a lot of phage therapy as well, which is a very niche and novel topic in the United States, mostly because it's very difficult to control and understand exactly what you're putting into that human.
So your regulatory approval is very, very slow on that front. I think veterinary uses are coming through first, because if you kill the occasional cow, you're going to get in less trouble.
Right. I mean, with the exception of El Java. At El Java, Eliava in the country of Georgia, and there's also they also do this in Poland. Um, you can get phage therapy, and they will essentially just continue to isolate novel phages or uh, um, stored phages that they have in their library until they find one that kills your strain of bacteria, and
Mhm.
then they'll just keep giving you phages until your infection goes away.
When you
And that's
think
pretty
it,
much the only way to make it work.
and that isn't that different to a doctor giving you five different antidepressants until your depression goes away, or
Exactly right.
the side effects aren't so horrific that you don't want to take it.
Exactly. And that's because at El Yada, oftentimes they have no idea what's in that phage in terms of its genome.
Mm.
And uh, so the one thing that I do caution about phage therapy is that um, it is very hard to tell the difference between um, what can sometimes be a temperate phage, which will form a lysogen, uh, and what is a virulent phage, which is obligately virulent and does not have the option to form a lysogen. And that's one of the main sticking points that you have to do a lot of due diligence in the United States to figure out that it is a virulent phage and not a lysogen, because lysogens are usually bad news because
Mm.
bacteriophages, I don't want people to think that bacteriophages are safe. Bacteriophages are very dangerous because lysogens are dangerous, and lysogens are uh, the combination of a temperate phage, which is living in the genome of a bacteria.
Mm.
Um, so let's take cholera, for example. Cholera, like that Vibrio, would not kill you if not for a filamentous phage that had infected it. And that's where that zonulocludens toxin comes from.
Yeah.
Sorry, that was way too deep, but I just know that um, I don't want someone to think that they can do some at-home phage therapy and that it's no risk,
Yeah.
because uh, phages are their own wily critters, and they seek to promote their own self-interest.
Hmm.
And if that means making that bacteria more successful, then sometimes they do that
Yeah. Yeah.
instead of killing it. And
Yeah.
so sometimes making it more successful and competitive means making it virulent or making it antibiotic resistant.
It,
And so
it's
we notice that a lot.
it's like hiring a hitman who the person you've sent it to kill, they outbid you and they send it back to shoot you instead.
Right. So, yeah, phage therapy can be useful. It also might kill you. So be careful,
Yeah.
because you can't always tell the difference between um, a, a temperate phage and a virulent phage,
Mm. Mm.
because um, the phenotypes can be very similar
Mm.
in terms of how the virus behaves with its host. And so that's when so one of the things I did, like I did the whole process of phage therapy, starting from phage hunting, so isolating novel viruses from the environment, to purifying them for injection into patients, and then like being the clinical microbiology lab that handles all the patient samples and figures out, um, did the phages get to the target? Were they growing? Were they killing anything? Was it effective?
Yeah.
Um, but I did a very similar um, sort of screen or selection one time where I took the patient's pathogen, um, and I was looking for a virus that would kill all the bacteria, even if the bacteria were not actively growing, and it should kill literally everything.
Hmm.
And so I took an entire liter of um, of this patient's bacteria that I grew in vitro, and then I spun all of that down after stationary phase. So all of the bacteria were just suspended in saline and they could not grow. And then I just kept flowing in phages until I found one that cleared the entire culture,
Hmm.
killed everything. And that I think is an important way that you have to really choose your setup carefully,
Mm.
so that your end result is actually what you want it to be.
Yeah, yeah.
Or else you'll end up with something else.
Yeah, 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 yeah. Now I want to make sure we spend plenty of time talking about how to lower the technological barriers to using microbes more as a tool. So, like in traditional food fermentation, you need to kind of clear the field. So you'll often do that with heat by like cooking the food beforehand, and you need to maintain those growth conditions only for the target microbes that you want to be growing there. You don't want it to get contaminated with other species that could be dangerous or just smell weird. Like there's all sorts of things that can go wrong if you don't control that fermentation. And like there's many very refined traditions for doing that reliably and safely. But what opportunities can you see for like our current wave of like cutting edge microbiology to feed back into those traditional cultures and change the way people do microbiology on a practical scale?
Um, I think if you're looking for stability and robustness and, resilience, then you have to transplant entire communities, and it's unlikely that you're going to see that kind of resilience with uh, an isolated strain,
Hmm.
especially one that has been um, spoiled in a lab.
Yeah.
And so like whenever I isolate strains, I refuse to spoil them.
Mm.
And so even though it takes me longer, I never grow them in their favorite media. I make them grow in conditions that they hate. And then I collect those and I put them in the freezer. And then, so one of Bactelium's longer aims is to actually genetically modify the entire uh, fermentation community so that it is robust and self-perpetuating. And so that, for me, it's a pretty simple system, right? I need Lucana stock, I need L. brevis and L. plantarum. I'm starting with L. plantarum because it's the most robust.
Mm.
But in the long term, that same trick of using an artificial chromosome um, will work in Lucana stock, and it'll also work in L. brevis. And I've already done it in L. brevis. I just don't have an artificial chromosome for it. And so it was intermittent production. But if we're going to take something like um, using the industrial process and the industrial tools that we have available, I would suggest that while we have them for now, we should make whole communities
Yeah.
that will produce these useful products and will be able to self-perpetuate for a very long time, because communities are stable and self-perpetuating for hundreds of years.
Yeah. Mutually
Like those
reinforcing.
right, kefir grains have been passed down from, for generations, and they are stable, they're fine, and they are safe. Um, but if you are trying to like pass down an isolate who has grown on, you know, the same broth for 1,000 generations or something like that, and then was put in a freezer and then was handed to you, well, you are inviting other microbes to come in and exploit that microbe's weakness. And so I would say that stick to whole communities. And sometimes you have to isolate novel communities,
Yeah.
which means finding one that you like and is not dangerous, but starting with a wild fermentation, so to speak.
I mean, this has clear parallels to the land racing movement or sheer total utter neglect from Mark Shepard. Like that
Mhm.
you have to be, it's very easy to ruin lineages of an organism by subjecting them to selection for conditions that you don't ultimately want them to perform in. I guess is
Right.
the way to put it. Yeah, yeah. Just a random thought. Do people ever do hybridization of these communities, either traditionally or modernly? Like take two Koji culture communities and like mix them together and then see what variations kind of come out of that combination?
Um, Anecdotally, yes, and scientifically, I know of one guy who that's kind of his entire life's work. He's in the UK, and he came to a conference that I was at back in 2022.
Yeah.
Um, so anecdotally, I have mixed a lot of different communities until I find what I really like,
Mm.
which for me is mostly something that is very, very robust. And so I've mixed together um, like some pretty robust kraut cultures with keeper grains, with um, some uh, scobies, and the result has been a really, really good uh, what's sourdough starter that I have that needs no babying
Yeah.
at all.
Yeah.
Um, and I took a bit of time to isolate some of the bacteria from it to find out what they are and sequence them, and the yeast. And so I ended up with a candida. So I ended up with the yeast from the kefir, but the bacteria that actually produces that, like um, the kefir grain itself, didn't make it.
Mm.
Um, but I have a lot of L. plantarum. I have a lot of L. rhamnosus. I have a lot of um, other lactic acid bacteria. And then it also forms a top SCOBY. Um, and yeah, I don't have to baby it. I don't have to feed it in between at all. And it always produces a robust rise, and the taste is great. The candida that I isolated out of it actually produces some awesome aromatic compounds. They're like they smell kind of like a tropical fruit.
Hmm. Wow.
So yeah, you can tinker with that stuff, and then you find what you
What
like.
works?
And if
Yeah.
you don't like it, that
So,
is your lab in your nose and your lab on your tongue that is telling you you
some,
probably shouldn't eat that.
yeah, there's something not right with this.
Um,
Do you think the emergence of scobies, like there's a few, if you look at the breadth of all traditional like microbial fermentations, scobies are a minority. Like it's really just kefir and kombucha that I know of. There might be some others that I've missed. But do you think
well,
that
there's
kind,
water kefir and
oh,
there's milk kefir,
you're right.
which are
Quite
completely separate.
distinct. Yeah.
Yeah.
But do you think that is a really good example of a practical technology that makes it much easier to handle those microbial communities, that you're not relying on a liquid culture of individual cells, that you can actually handle a relatively pure self-reinforcing lump of microbes between batches?
I think it's very helpful.
Mm,
Yeah, but not always necessary. Um, it's convenient, but not required, I
mm,
would say.
mm,
Mostly because there are a lot of ways to catch a whole community,
mm,
right? Um, you could probably just have some fired ceramic beads that you have in your fermentation, and transfer the beads from fermentation to fermentation, and that will pick up the entire community and take it to the next fermentation.
yeah,
And
yeah, yeah, yeah.
so you really don't have to um, you don't have to go high tech with it. And again, I am finding so many situations where just regular fired ceramic in your backyard, like low heat fired ceramic, is one of the most useful things
Mm,
when it comes to microbiology.
hmm hmm
Um, so that, you can be creative if you can, but again, always just make sure that it smells and tastes like something you want to eat
Yeah.
as a result. Um, but yeah, if it doesn't produce a SCOBY, you can just you can provide it a home.
Are there any other ways of preparing feedstock for fermentation other than heat, which just presumably kills everything that isn't heat tolerant, or salinity, which means you have to have a source of salt, which if you live by the beach is great, but if you're inland, it's an input that you have to negotiate where you're getting it from, or pH. I guess there's a small number of like alkaline, high pH controlled fermentations, but they usually tend to be low pH acidity. Are they really the only ways of controlling contamination?
Well, the only other one that I have ever used is filtration,
Ah, yes.
right? And so in that case, that brings me right back to ceramic because you
Interesting.
can filter, yeah.
And you can do that under gravity if you get the scale right too, because then the weight of the water column will make it work at a reasonable rate.
Yep.
Interesting.
Or if you're just patient, it'll weep out because of capillary action.
hmm
So
hmm
that's the other thing that's really great about ceramic is that it doesn't clog. Like the filter doesn't clog because even though you may have like a layer of scuzz that builds up at the bottom of your vessel, as long as it is still wet, capillary action will actually just continue to drive moisture through the ceramic, and but then you're just going to weep, right? Your flow is going to slow down, but it won't stop. Whereas like all of your modern filters, like nitrocellulose or um, polypropylene or something like that, like they're going to give up the ghost as soon as they get clogged, and
Mm,
then they're just going to rupture.
mm.
And so, uh, yeah, that's the only other method that I have ever used, and that only works on liquid stuff, right?
Yeah.
But you can protect your thing um, inside of a ceramic vessel, and you can feed it from outside of the vessel uh, by just setting your vessel in a crock of whatever contaminated stuff that has the nutrients that you want.
Yeah.
And
Yeah.
then you can grow your pure, not pure culture, but you can grow your uncontaminated stuff inside of your ceramic vessel. And ceramic vessels are also great um, because, well, you can remove all the organics by burning them.
Yeah. 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 Yeah.
So you don't have to have any special kind of sanitation equipment. All you can do is just refire them at low heat.
Yeah.
Um, and then you can remove all the organics and you will kill everything,
Mm.
even the spores.
Has anyone looked at so with low pH, like the sauerkraut kind of ferments, you normally start with a big batch of material, or the salt kind of helps with that too. But let's a hypothetical ferment which is controlled just by low pH and not salt. Could you do it more as a flow production rather than batch? So you don't have that early phase where there's a small amount of the target desirable microbes struggling like mad to get the pH down quickly and prevent anything else growing. So instead, what if you had a large batch that had already fermented, was stable, had only desirable species, it was acid, and rather than taking a small amount of that culture out, you took out maybe 20% of it, added 20% more feedstock, and you don't have to like sterilize that feedstock because you have all of that low acidity microbial community waiting to ferment it rapidly and drop the pH before anything else can start to become a significant factor.
So that's a chemostat. And so a
Ah,
chemostat, right.
yeah.
So that's used in industrial microbiology all the time.
Hmm.
At my postdoc lab, we had several of them running all the time for the methanotropic bacteria. Um, though the problem with methanotropic bacteria and chemostats is that they're highly susceptible to contamination because they're carbon fixers and
Yeah.
they are not changing the pH and they're producing a ton of sugars and amino acids that other things would love to eat. And so something will show up and eat them. But, you know, what I like about ceramic is that to, um, you don't have to remove anything. You can just flow it through the ceramic, which sterilizes it,
Yeah.
and you can just let osmosis bring new nutrients in to your fermentation vessel. Now, if you're using inedible plant parts, then you're going to have to remove the plant parts after a certain amount of time and
Mm.
then put in fresh ones. But you don't have to change any of the liquid and you can leave all of the microbes in place for the most part.
Yeah. Yeah.
Um, so that's my preference for a chemostat is that you don't have to um, remove like 20% of the total volume because for me, the biomass is not the product, right? And since the biomass is not the product, like the biomass specifically meaning the L. plantarum, the pickle bacteria that I'm using for this fermentation is not the product. It produces the products that I want to have.
Yeah. It's the catalyst.
Um, right. And so I want to leave all of them in place and I just want to keep adding new feedstock
Mm.
because if I remove 20% of them, I would then need to spin down with a centrifuge that bacteria in order to harvest it. But in this case, I don't have to do that.
Mm.
And so the allowing seawater specifically to flow through will one make the fermentation last much longer because of the high pH, the alkalinity of seawater, and it'll also bring in more minerals.
Mm.
And so when I was listening to your talk with Steve Solomon, you know, nothing is ever zero input, but you can get pretty close.
Mm.
And as close as you can get is when you're down to the minerals and salts.
Yeah.
Um, and in inland places or in leached soils, they may not be present, and you can't escape your need um, for iron if you want to have hemoglobin and myoglobin
Yeah.
be functional, right?
Yeah. Yeah.
Or if you want a if you want an enzyme to work, it's gonna need some divalent cations, and it's probably going to need some metal
Mm.
at the active site for that enzyme to have its catalytic effect. And so you just need the minerals. But the thing with the thing that ocean water allows you to do is it allows you to continue to flow in those minerals, which are dissolved in ocean water. And there's a lot of minerals dissolved
Mm.
in ocean water. So you can escape having to mine for those things.
Do you suspect that there's a potential synergy between crop breeding and microbiology that hasn't been fully exploited? And like the example I would have, I'm looking at a big pile of sword beans, which have a fairly nasty alkaloid in them. So the simple way of preparing them is just to do multiple changes of water, but that means that you need more firewood to prepare that for human consumption. But as a contrast, if you had the right microbe, that would be a nitrogen source, and it would happily chew through all of that alkaloid and give you an edible product without having to use a mountain of firewood. So I don't know, or even like biomass plants that are nowhere near edible for humans, but are suitable, like they're bred and selected to be compatible with a fermentation system. Have you had any thoughts about like that possibility emerging in the future?
Um, so for me in particular, I'm interested in inulin, which is not digestible by humans, but it's highly soluble and it's a great carbon source for L. plantarum. And so I have some genes that I stole from Aspergillus um, that will allow it to secrete an enzyme that will break that down, the inulin down to fructose. And so that way I can use like the Asteraceae plants that produce a ton of inulin. When it comes to toxic compounds, it is almost certain that the enzymes exist.
Mm.
Um, whether or not they have been characterized and someone has been able to successfully isolate and clone that enzyme from whatever microbe can do that, that is just a question of time and resources. It's not really a question of reality
Mm.
because of how deep the bank of biodiversity is in the microbial world. Um, so, right, it would be more unlikely that there isn't an enzyme that can break that down.
If
The
someone out there
question
is
and
looking, if someone out there's looking for a PhD project, I reckon finding a practical fermentation system for breaking down tannins for oak detoxification, rather than trying to breed oaks to stop making tannin, that I reckon would be a winner.
I think so too.
Mm.
And so um, I think that's a much more productive use of time because it's so much faster and you'll have a result much faster that could last again essentially forever, right?
Yeah.
Because people have been passing down these fermentations
And
for
the
generations, who knows
tannins are
how long.
useful. The tannins are useful. It makes the crop easier to grow and manage and harvest. It just makes it difficult to do the last step of actually eating it. So you just need a microbial community to help you with that final step. So you don't need a running stream and days of like water running over the cooked acorns.
Right. Now the problem with that is you go and you find a microbe that has that ability in nature. I'm sure there's one in a squirrel's gut,
Hmm.
right? And I'm almost guaranteed you could probably isolate that in a matter of months, and you can have a tannin digesting microbe in almost no time if you go looking for it. But the problem is, would you want to eat it? Because you don't know, it could be something that would be toxic to you.
Hmm.
It could be that that microbe is something that is just gross. It might produce a bunch of polyamines and it might putrefy your acorns. And so that's kind of why I like the path of finding an enzyme and transferring it to something that you already know is safe and delicious.
Hmm.
Because, well, you never know what you're going to get when you go looking for wild microbes. It might be something that you love, or it might be something that'll kill you. And so you have to be careful about that. And it depends on how much time you have to go looking.
Mm.
Um, and so if you are trying to, let's say, like utilize as much of Frankenstein's toolbox as possible before it goes away, then yeah, that would be the tack that I would take is to just go and look at the genetics of those microbes, find the enzyme, transfer it to something that you know you are fine to eat, and make sure that that enzyme can be persistent, which means growing that microbe on tannins
Mm.
pretty much all the time, and don't let it get soft because they'll just if you take away microbial genomes will cut out stuff that they don't need
Mm.
if you let them.
Yeah, yeah, yeah. They need that continual incentive. Otherwise, they'll find an easier way.
Mm-hmm. So yeah, I think that's almost guaranteed that that would be something that a single PhD student could accomplish during their PhD
Yeah. Yeah.
because of how much genetic information is already available. I would be surprised if someone has not done a squirrel microbiome, analysis already. I would be surprised if there's not a metagenome where someone can go and like pull a genome of like a crappy genome, but a genome
Yeah.
of the like a bunch of different bacteria that are in a squirrel's gut.
Yeah. Yeah. The alternative is to domesticate the squirrels and then just eat them. Let them deal with the toxin, and we'll have lovely squirrel, nice fat ones that just come straight to you.
Have you eaten squirrel before?
I haven't. I haven't been to the US. I've only seen them once in London.
Oh, they're quite good.
Mm.
I have squirrel and dumplings. One of my grandparents would make that
Oh, there you
for me.
go.
It's quite good.
Yeah.
Yeah, yeah. Squirrels are delicious, but they do have a habit of carrying tularemia. So
Hmm.
you got to be careful.
Yep.
That's
Yep.
one single bacteria is enough to be a lethal dose for a human. So you
Yep.
got to be careful with
Hmm.
that.
Did you want to
Sorry, that's
shift?
the microbiologist in me.
Yeah. Did you want to shift on and if you, I think we've covered a lot on that aspect of like lowering the barriers and enhancing the longevity of microbiology as a science for the whole human
Yeah.
race. Did you want to talk a bit about uh, networkings and communities and like how to, I mean, maybe even some advice on like if people are interested in starting to play around with microbiology more? They might have done some sourdough fermentation, but this is like the next step and the next step to start doing more research oriented approaches. What kind of community is out there? How do people engage with it? Where do you source resources and materials and advice? Like, how does that all work? I don't know very much about this, part of the world.
Um, so there's a lot of tinkerers out there, and it's mostly underground, and mostly everyone is really quite well behaved, which is why we haven't been cracked down on at all, and there aren't many regulations on it. And so, as long as you are someone with good intentions and you are like intend to be well behaved, then I would say, come join the community. And most of the stuff that you need, you can buy on Amazon or you can buy in a grocery store. But it does require um, knowledge, but that knowledge is actually on the internet. And you don't necessarily need to get a degree or something like that
Mm,
because um, you can volunteer in labs, at least in the United States. And when you're a volunteer in that lab, you can learn some of the skills that you might need, but the actual resources required, um, you can pretty much all buy on Amazon or at a grocery store. Like, for example, the Epsom salt and Miralax, which is the brand name of polyethylene glycol.
mm,
Um, those you can buy at a grocery store, and those allow you to transform uh, bacteria at home
hmm
pretty easily, which is pretty scary when you think about it because those are the most commonly used laxatives. So those are added to the human gut all the time. And so that is certainly facilitating horizontal gene transfer, which is sometimes good and sometimes bad, um, depending on, how much luck you have. So if you're lucky, it's good. If you're not, it's bad. So, just a microbiologist word of wisdom, try to eat more fiber rather than relying on uh, commercial laxatives or stool softeners if you can. Um, and then Adgene. So there are several nonprofits who make both strains and the genetic materials available to researchers. So this is where you have to volunteer in a lab. You cannot gain access to a plasmid on your own. They will not ship you a plasmid to your home or something like that. But you can you can volunteer at a lab, and when they know you, they might let you order a plasmid that you're interested in, and work on that.
Mm,
And then sometimes things get taken home.
mm,
Um, but that's kind of how the community is working right now, because though it's not illegal to ship those plasmids to someone's home, it's an industrial standard that no one does it. Um, so if you're going to get them, it has to be something that you either source from someone else in the community. And again, they're only going to send you something if they have pretty good evidence that you are going to be well behaved. Because again, this is mostly a self-regulated community.
mhm.
Um, and then you have the route where you can just volunteer in a proper lab. And volunteering might mean that you show up like uh, once every other week, and putter around in the lab a little bit and wash dishes or something like that.
Mhm.
Um,
I guess is
Depends,
it,
and you can
it
just write them an email and see if they'll let you show up and work and learn.
is there another approach to take to start in the like the traditional fermentation community? People like learning how to make natto and koji and all those kind of things. And I think wild ferment is a growing tradition as well too, to source, all of the biodiversity and genetic diversity that's in the environment around you that can be added in to those wild ferments, which isn't always 100% safe. Occasionally things end up in the mixture that
Mhm.
could have negative consequences. Like disclaimer, like any of this kind of stuff is, but then again, people occasionally grow zucchinis that like revert back to bitter forms and end up dead from eating poisonous vegetables. So like this is life is potent, and there's always a chance of something
Right,
unexpected and dangerous happening. And there's things that you can do if you learn through the community to minimize the chances of that happening.
right. So if you're interested in uh, just isolating microbes from the wild, and starting your own library of interesting bacteria, then that setup is much easier to do, because you can get petri dishes, or you can get media and stuff like that, or you can make your own media um, at home, because you can buy glass petri dishes and you just wash them. And you can use a pressure cooker as your autoclave, and you can sterilize your stuff if in case you need to. Um, and yeah, you can make your own media with agar that you buy for food, right? Like if you have vegetarian jello, you can use that to grow a lot of bacteria.
Mm,
Um, and then when you're going and looking for wild things, um, I would say that your nose knows for the most part. Um, And then it wouldn't hurt you to buy a pack of pH strips, because there's a lot of, like, you can just buy a pack of pH strips. It's like $5 or $6, at least in the US, on Amazon. And if it's below 4.6, if you're doing an acid fermentation, then it's generally safe, because Clostridium is not able to grow below that
mm.
pH. Um, and so you're safe from botulism. So keep an eye on pH, but besides that, just make sure that it smells like something you want to eat. And if
Mm.
it doesn't, don't put it in your mouth. Um,
I'm guessing there's overlaps with the wild foraging community. Like there's a few very deadly mushrooms that you have to know to identify, and there's some lookalikes that aren't worth taking the risk with. But then everything else is from delicious to will give you a stomach ache for a week. And you can
yeah,
have strategies for how you begin consuming that to minimize the risk of anything really bad happening.
right. Like I'm doing uh, because I'm interested in L. plantarum. I'm doing a wild fermentation right now. Uh, we took my daughter to the beach about a week ago, and then I saw some bull kelp on the beach, and I decided, well, I know that L. plantarum also lives in the ocean, both at the top on floating algae, and also like in the sediments of the deep ocean. and it's also pretty much all over land. L. plantarum is everywhere.
Mm,
But I was like, well, maybe I want an ocean one. Maybe I want one that is adapted to growing on seaweed in case I want to try and ferment seaweed. And so right now I have a very acidic crock of bull kelp, and I did nothing but add seawater, add bull kelp, and now I'm going to use that to isolate um, some other strains of lactic acid bacteria that eventually I might transform
mm,
um, as a part of this project. But again, like you don't have to do the transformation. You can just, as long as it's a low pH, it's nice and acidic. You might just find something that produces a novel aromatic, like my, tropical fruit tasting and smelling um, yeast, right?
mm,
It's really common to get banana. Banana is really easy to get with yeast, but like this really fruity, almost Jolly Rancher um, smell and taste is something I haven't ran across since. And so you only get that when you go exploring.
mhm.
So if you're looking to explore and do wild stuff, I would say the part of that is helpful and useful is finding new flavors and smells that enhance your cooking.
Mm. I'm going to have to pick your brain off air if you have any other people in this community that you recommend I track down for interviews, because I want to do more of this. I think it's a really important ingredient into the toolkit. Oh, did you have any final thoughts that you didn't get to? I know you said you had a couple of notes about reading Taming the Apocalypse.
I do. So when I was reading your section on animals that would be sort of good candidates for novel domestication, I saw uh, in the terrestrial crustacean section a sort of a hole there that I've had a plan to try and fill myself for like 4 or 5 years now. I just haven't gotten around to it, which is the woodlouse.
Oh, interesting.
Right? So the woodlouse is a true crustacean. It lives for real on land its entire life, where those others have a stage of their life where they need to be in the water. But
Mm.
you can be completely inland and grow a woodlouse on terrible material, and they'll be perfectly happy. They just need a bit of moisture.
They've got
It
really
just has to be
effective
damp.
they digest cellulose really efficiently, don't they?
They do, yeah. So they are an excellent candidate for like waste material. But the thing is, they're so tiny, right? And you're not necessarily going to want to eat all of that shell. And, maybe you could grind them up and feed them to something else, but then you're like wasting your trophic effort, so to speak. You are
Hmm.
like adding another level of, like, um, you're adding another trophic level, which decreases your efficiency.
Mm.
And so I have a plan to actually make a much larger woodlouse through traditional breeding, but after um, a transgenic enhancement. And so, like, one of the main limiting factors is oxygen because they suck at circulating and gathering oxygen from the atmosphere and circulating it through their body. And so, like, they just have like kind of one big muscle mass in the middle. Um, And my goal is to transfer myoglobin to that muscle mass so that it can retain oxygen and make it more efficient, meaning you can get them a lot bigger through traditional breeding. But because their generations are so short, in
Mm.
like maybe like 10 to 15 years, you could probably have like a lobster sized woodlouse as long as it has the ability to hold onto oxygen more efficiently
Yeah, yeah.
growing
Okay.
on the land.
Yeah, yeah. I
So,
love that. I love that. I mean, I'm sure I've seen some in the fancy pet trade, the exotic pet trade, that are quite large. They're like maybe an inch
yeah,
long.
yeah, they can be about the size of your thumb.
Yeah. I mean, people peel shrimp that size, so I wonder what they taste
That's a
like.
lot of work. Uh, the taste can be highly variable, so you wouldn't
Hmm.
always want to eat them. But that would be something that I would select for um, during the traditional breeding process.
I love it. Do they have their own gut microbiome, which could also be tweaked?
Yes, yeah,
Hmm. I could almost
I
see them
think,
working with like a symbiotic fungus to break down, almost like a termite colony, but that humans manage with edible uh, pill bugs, slaters
yeah,
instead.
yeah, I think the to me it sounds like it would be a really good way to make sure that people have a good source of protein on
Hmm.
land
Hmm.
inland. Um,
I love it. I love it. I think that counts as your, if you could dream up any organism that you'd breed. But have you got any others on that list? Because I'm sure that you have more than one.
uh, for sure. So if I had a magic wand, or if I just had uh, like a long enough life and like enough resources, I think the first thing I would do is make a cold hardy universal interstem.
Hmm. Hmm.
I think that would be the most impactful scientific project that like humanity could dream up. Um, because you could, right, you could, if you had a universal interstem, then you could essentially just pop that in between a mesquite tree in the desert and whatever fruit you wanted or whatever vegetable you wanted, right? You could turn a mesquite tree into, a big tomato producer,
Yeah.
right?
Yeah.
And they have like the longest tap roots on earth. They can reach like 200 feet down to the water table, which would allow, those trees to persist for multiple generations, even if the drought lasted, multiple generations because they're directly in the water table. Um, but it would also allow you to graft onto mangroves,
Yeah. Yeah.
um, which would allow you to then use the mangroves to produce whatever you want.
Mm. Mm.
And so you don't need fresh water again. Um,
That one's amazing. And
and then,
there's some research that reminds me. They found that Solanaceae, the tomato and potato family, they produce enzymes during graft healing that seem to allow them to be compatible with almost anything. They can reform the tissue of the plant that they're grafted onto. So there's hints of that potential hiding in the plant kingdom. Just somebody
yeah,
needs to tease it out.
yeah, I did read a paper one time about someone, they had modified a tree tobacco. I think it was like Nicotiniana, maybe it was Bentha. I'm not sure, um, but then they were able to do, and I think it was Asteraceae to Solanaceae. Like they were able to do like a graft between two distinct families.
Yeah. 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 Interesting.
But I think that would be really helpful, but it would need to be cold hardy, I
Yeah.
think,
Yeah.
which of course Nicotiniana is like mostly not cold hardy.
Mm.
Um, so, and then if I could make one more, um, it would be a mesquite tree that doesn't produce so much sucrose, instead produces starch
Hmm.
and also produces gluten. And then it would have an inducible masting cycle.
Yeah. Oh my God.
And so,
Oh, that
right?
is perfect. And
Yeah.
are the fruit, they'd either dry spontaneously on the tree or
Mhm.
they'd be storable in like their complete form.
Yeah. Yeah. The mesquite tree I've been pretty obsessed with for a long time. Um, but I figured that in my lifetime, I won't be able to do that because I would have to figure out how to make protoplast, and then I would have to figure out how to bring uh, the mesquite into culture, and then see if I could finally make a genetic modification. But then I realized that I actually ran into an enzyme by serendipity, um, working with that methanotrophic bacteria, and it's called amylosucrase, and uh, it converts sucrose into not quite starch, maybe like because like you might get maltose or something like that, but it makes polymers of glucose, and then it targets the fructose for reutilization, like
Mm.
towards fructose 1 phosphate, and then like until it becomes glucose again, like fructose 6 phosphate, and then isomerized back to glucose.
Mm.
So yeah, and so my plan for that is actually just to um, add that to my fermentation arsenal so that people in a much shorter period of time could really turn mesquite into a useful
Into a staple
crop,
crop. Yeah. Yeah.
into a staple crop, because you could ferment it, and in the fermentation you could convert that sucrose into starch, leave a lot of your calories in place, and then because it's so nitrogen rich, add some gluten, because I already have a cassette for getting the bacteria to produce gluten.
Yeah. Yeah.
And so you can essentially have bread in the
Just
driest desert
just
for decades or generations on end.
just hanging off the tree.
Yeah. But I
Brilliant.
don't know how to do the inducible mast cycle. That would be great.
Yeah, control of flowering in plants is still a bit of a mystery. They're starting to make progress on what the triggers and the cycles are, but I mean, particularly if it's inducible, because if you have like a pest population building up in a region, you're just like, we're just going to not produce any of the crop this year, and the
Mm-hmm.
pest population has to crash to
Right.
compensate. Yeah, that is brilliant. I'm so glad I got you on. Oh,
Yeah, I'm glad to be here.
I want to, I so rarely get to talk about these kind of things. I want to throw an idea at you. Do you think
Uh-huh.
there are any obscure carbohydrate sources in particularly the plant kingdom, just because they're easier to grow and scale, that could be converted into a human food by changing the human gut microbiome? That we could actually develop a gut symbiote that allows us to eat something that's like even weirder and less digestible than inulin. And that makes those crops really easy to grow because there's nothing in the ecosystem other than humans that know. I guess you'd risk that microbe ending up in the guts of the rats and the mice soon enough if it's in all the humans.
If it isn't already, right?
Yeah,
Because that's probably where you would go looking in the first place.
yeah.
Is you would go looking uh, in the guts of something that eats that routinely.
Mm.
Um, I think that so it would require a bit of metabolic engineering, right? Because the thing with inulin, right, is that if we're taking that as an example, is that you already have microbes in your gut that can digest inulin.
Mm.
But the problem is that they are producing usually lactic acid and um, a lot of gas.
Yeah,
And that is the problem that's leading to constipation and diarrhea and a ton of flatulence, which is why people don't eat it. You would need to have, um, you would need to also make sure that it has a metabolism that produces less gas and something a bit more neutral and less of it. Maybe some short chain fatty acids would
hmm
be preferred that can be absorbed, something like propionate. Um, but that's the much safer route is if you have a microbe that will produce a short chain fatty acid that
yeah.
is actually caloric to the human. And then you're essentially turning your guts into a
A
fermenter
fermentation
that
tank, yeah. The
yeah,
thing I think would have the most potential with this would be some species of algae because they've got really weird carbohydrates in them.
so many weird ones. Yeah.
Mm.
But I mean, there's no end to weird carbohydrates. Like
Mm.
the more you go looking, the more you find. And the thing is, the looking is not so easy.
Yeah,
So,
yeah.
because like the bigger they get, the more they're going to clog up a column.
hmm
And like then what are you going to do?
mm
Um, in order to like resolve that structure. So yeah, I think in like the current iteration of human civilization where we are right now, I don't think we need any more calories.
Mm.
Um, and I think it's probably generally better to do that fermentation outside of the human. But in the scenario that comes up in your book, which is when you want people to survive the collapse of an industrial civilization, or more, that sounds so catastrophic, reversion to the mean
Mm.
of human existence, right? Like if we have a reversion to the mean, um, then that's probably something you would want, right? Because you want to be able to extract as many calories as possible.
Mm.
Um, but along that vein, if you're already doing some genetic modification and you're adding things to humans, I think maybe you should just add a new uh, endosymbiont,
Yeah.
right?
Yeah.
Like you could probably also like there's no reason that you could not genetically modify a chloroplast such that it would behave properly in a human cell. And
Hmm.
then as long as it's in the epidermis, it'll get enough light that you can produce some useful carbon compounds and some extra oxygen. Though, who knows how that'll turn out? Probably bad for a while, and then eventually would stabilize.
Mm. Mm.
I don't know. I mean, if we're talking because a chloroplast is essentially just a bacteria that happens to live inside of the cell instead of outside of the cell.
I just realized too, I've seen estimates about how much useful energy a human with chlorophyll in their skin could capture, but that probably doesn't allow for the fact that they'd be operating under a higher effective carbon dioxide concentration. That's the real limiter. It's not the photosynthetic efficiency. It's just knowing what to do
Mm-hmm.
with that energy. So
Yeah.
I wonder if that would change the calculation. Anyway, that's um, I did want a final question that I want to throw at you, and I think
Uh-huh.
this is the most provocative thing in Taming the Apocalypse, and you've got more hands-on experience with this kind of stuff. So my
Mm-hmm.
theory is that biological technology, particularly genetic manipulation, has the potential to outlast the end of the industrial age. So when we no longer have
Mm-hmm.
supercomputers and CRISPR and pure reagents that turn up in the post from a factory on the other side of the planet, that nevertheless we still could maintain some kind of culture that catalyzes genetic rearrangements on all sorts of different levels of the ecosystem and to be involved in that process of tinkering and then selection. And I'm curious what your thoughts are on that. Can you see a low-tech genetic engineering revolution carrying on into the distant future?
I mean, nothing is impossible. And so I try not to make predictions because every prediction is going to be wrong in some respect. But sometimes your predictions are right enough to be useful.
Mm.
And so I would say that when you get down to it, molecular biology could be done as an apprenticeship. Like one of my life projects that is hiding, back in the secret part of my heart is I want to take an apprentice someday, and I would want to train an apprentice in molecular biology and have them be able to do the things that I can do with almost no technology, with a low budget, because of the knowledge transfer. Because there, when I went through the PhD, so obviously from my background, higher level education is not a thing, right? Like even literacy is kind of like questionable. Um, so what what does work though is like the apprenticeship system,
Mm.
hands-on learning. And what I saw when I was in the PhD was essentially it's as if a bunch of smart people who had too much money and not enough good sense recreated a worse version of an apprenticeship.
I
And
love
that's
that.
essentially what a PhD is.
Yeah.
And so it's all backwards.
Mm.
And I think that there's, and whenever I bring up that topic, I get a lot of pushback because they think that, well, then they'll only know how to do things and then they won't know how they work. And I was like, well, that's that's only if the person training the apprentice doesn't teach them that,
Yeah.
because there's, you know, uh, a lot of people, they have theoretical portions and reading and like in-depth conversations with the journeyman that's training them, and that can easily compensate for the classroom learning, which I'll be honest, I was really not impressed with the retention on
Mm.
that. So I ran into people who just, so my background is not a scientific background, but I ran into a ton of people who had, 4.0 GPAs and double majored in biology and chemistry who don't know what soap is, right? And they don't know how to make soap. They don't know that, soap is the salt of a fatty acid. You neutralize a fatty acid and then you get soap, and it's different from a detergent, but that is something that, you know, uh, your ancestors didn't know necessarily that that's what they're doing. But if you know that, you can impart that onto the next generation.
Yeah.
And so I think that there's no reason that the fall of access to recorded knowledge should result in a fall in the perpetuation of that knowledge,
Yeah.
because
Yeah.
it's no different than becoming an electrician. It's no
Yeah.
different than being a plumber. It's no different than being a mason. There's a lot of skill that you have to have, and there's a lot of knowledge too in order to do it well. And the quality depends on who taught you.
And I think it's a tragedy that there are so many exceptional people such as yourself who would make excellent scientists, but they would be terrible at doing exams for 4 years to get the privilege of, having a try at doing a
Mm-hmm.
PhD. And I also realized too, what a PhD kind of is like is similar to what you're doing with your spirulina. You're putting it in a
Mm-hmm.
difficult, constrained, but protected situation where it's got this,
Mm-hmm.
unlimited flow of information where it can cobble various elements together and try and find solutions to that problem that just won't go away.
Right.
Mm,
So, yeah, I don't see any particular reason why it would have to stop. And I
yeah.
think that entirely depends on us because, okay, say, right, if you want to do a DNA extraction, um, right now, you're pretty much always going to use SDS, right? Your sodium dodecyl sulfate. That's the commercially available detergent because it's so cheap and available and it's everywhere. But there's no reason that you can't break that membrane with lye or with lye soap.
Mm,
That will disrupt a membrane just as well, and you can get at your DNA. So it's about how clever you are and how well you can transmit that to the next generation,
mm,
is what I think.
mm.
And so I think that um, there would be no reason for it to stop unless, well, the thing is, a lot of scientists, I feel, are very uncomfortable with that dynamic,
Mm.
accepting the responsibility for the stewardship of their own profession. And I think that's an area for growth um, in the field. And I hope that happens uh, before your scenario finds its way into our lives.
An analogy I would maybe make was with the end of the classical period in history. There was all of those libraries and these very official repositories of all the knowledge of the day. And they were all burnt down and looted when things fell apart. It was little monasteries out of the way that managed to keep fragments of things that were useful or interesting and to learn and to invest the energy into replicating them and sharing and spreading them. It was that network model of
Mhm.
information that ultimately survived, not the Library of Alexandria, where, it had everything for like a few hundred years.
Right. And so, if I have enough time, someday I will start an apprenticeship program for scientists.
Mm.
Uh, but God, scientists are like herding cats. Like if you even bring up the topic of organization to scientists, they oftentimes get very surly and they want to do their own thing because oftentimes, like if you're going to run an apprenticeship program and set standards and stuff like that, you're going to need a union or a guild, right?
Hmm.
And those are functionally like just different interpretations of the same thing, right?
Hmm.
You're promoting an industry or a class of artisans, and making sure they can survive in the economy. Um, but yeah, trying to get scientists to organize and contribute to a common goal, in my experience, has been very difficult because they are unwilling to be persuaded.
Right now in history, it feels biotechnology getting out of academia into the general population feels like the early days of like formal science when it was gentlemen
Mhm.
scientists who had like, time spare and a bit of money to splash on equipment and societies of letters. It was mostly people like writing letters to each other. Oh, I did this weird experiment the other
Mhm.
day. Look at this crazy thing I found. And they were like just kind of having fun and showing off to each other and motivated by curiosity. That's kind of the developmental stage that it feels like backyard biotechnology is at. And given time,
Right.
I think the guild apprenticeship formalization is probably eventually going to emerge. I know it's just such a fascinating thing to watch it kind of bubbling up.
I really hope so, because the way it turned out the last time was not great, right? I mean, that was uh, like Humboldt. Humboldt was one of those. But he was an aristocrat and he played into the expectations of the aristocracy and academia at the time, which was that he suffered from having to pay for his own publications. And so, you know, he was struggling to get enough money for his experiments, for his trips. And then when he had it, he had to spend it on publishing, whereas other people, right, they get paid to publish
Yeah,
things,
yeah.
especially because his books actually sold quite well.
Mhm.
So, mostly because of the illustrations, but um, you know, the gentleman scientist, I think, is uh, it's certainly possible now because the scientific instruments are cheap and there's a bunch of industrial excess. You can go on eBay and you can buy a centrifuge for like $150.
Hmm.
You can set yourself up to do PCR. You can set yourself up to do electrophoresis for cheap.
Mm.
Um, and so you can do a lot of experiments on your own, and you can, gain some knowledge. But yeah, the thing about writing letters to each other is kind of what we did right here.
Yeah, yeah. The internet is the new format where all of that is kind of bleeding out.
Yeah,
But I think it still has a curation and a signal to noise issue, and even a searchability issue.
yeah,
Like Google doesn't really work anymore. Whether AI is going to fill that gap as a de facto search engine, maybe, maybe not.
yeah. The world was just smaller back then.
Mm.
Like even if you read papers from like the 20s, like Crabtree when he was writing about um, uh, aerobic fermentation, it read pretty much exactly like a correspondence, like a letter. And he was essentially writing that to Warburg in, uh, um, in what would eventually become Germany, right?
Mm,
So he essentially had an audience of one, but it was made in a mass-ish publication. Um, and that's kind of like, I think Substack can help get us there,
mm,
right? You can make it available for everyone, but then if you write it clever enough, then you know that you can get an algorithm to serve it to your, audience of interest.
mm, mm. That's definitely what I'm
But who
trying to
knows
do
how
on.
long that'll last?
Yeah, that's definitely what I'm trying to do on Substack. It's basically a recruitment drive to get more people interested in breeding crops, livestock, and microbes as well. I think now's the time in history where anybody with a little bit of spare time and resources could invent something that lasts for 1,000 years and that benefits society along
Yeah,
the way.
yeah. Though I do from your book, I think that the Colossus may not need to fall.
Mm.
It may just need a crutch.
Hmm. Hmm.
And
Yeah,
so if
yeah.
I can get systems like, or a prosthetic,
Mm.
um, one leg may be going bad, but we might be able to just cut it off and put a new one on, as long as it, doesn't uh, it doesn't fall over during the adjustment period. Then maybe it could work. And I think that precision fermentation could be a crutch
Mm.
because of the level of efficiency. Um, and right, it's just highly efficient. Or it can be, because microbes can be the direct beneficiary of their own nitrogen fixation or
Yeah,
their own carbon fixation.
yeah, yeah.
Um, and you can grow them in salt water, and the salt water can have all of those micronutrients that on land would require trains, trucks, and ships to bring you those micronutrients, and you would have to spread them out carefully. And then again, most of them are going to end up in enzymes in the rest of the plant, not the food that you eat. So
Mm.
again, another efficiency loss.
Mm. Yeah. No, it's exciting times, and I'm extremely grateful that I got a chance to not just talk to you, but share you with the audience as well too, and hopefully put a call out there as well if there's other people in this broad field of hands-on microbiology. I need to talk to more people in this community. So with that.
Yeah,
Uh,
I may be able to find you some.
yeah. Let me know. But yeah, I think we have to wrap this up. It's been a while. It's been a long episode, but it's been a great one. So, Dr. Willkeen, are there places that people can follow up with your work? I'm guessing Substack with Bactelium is probably the easiest place to read some of your articles.
Right. You can find me on Substack through Bactelium, but we also have a website where you can find links to both our Substack, which I'm treating as a sort of open source digital lab notebook. So as I conduct experiments, I'm going to be updating all of the different product or the different projects that I've already introduced. So the current projects are starchy squash, the production of casein in L. plantarum, ovalbumin, that's egg white from hens, myoglobin, that's like the meaty protein from beef, gluten from wheat, and starch also from wheat. Um, and so as I make progress in each one of those, you'll be able to read what the result of the latest experiment was. And I'll try to post pictures. But if you go to our website, you'll also find links to our Give Butter, which is our donation platform, because we are a nonprofit research organization. And so we need donations in order to pay for the things that make this work. Um, so in case you are interested, read. In case you are feeling generous, donate or maybe do both. But yeah, Bactelium is going to keep working on finding a crutch for the colossus of the industrial agricultural system, meaning that we could save ourselves from a crop failure because you could ramp up a fermentation in a week, and it could be done in a month.
Yeah, yeah.
And then you could, right, like, so it's rapidly scaling, and it's not sensitive to climate catastrophe. If anything, warm ocean water will actually only make the fermentation happen faster.
Well, there's nothing more adaptable, flexible, and scalable than microbes. So I think
Right.
you might be onto something there. You've challenged my position. I'm like, I think there's a chance you'll be right, and I'm hopeful that things will work out, and that yeah, I'm wishing well that people like you will be contributing to that solution. So I think we'll wrap it up there. And maybe I'll get you
Sounds good.
back on in the future because I would love to share some more updates of all of these amazing projects.
Yeah, I'll be sure to let you know in particular whenever I do something exciting.
Yeah, brilliant. Okay. Well,
Yeah.
thank you very much.
Thank you for having me. And yeah, hopefully someone in your audience enjoys what I'm doing and wants to check it out too.
The Zero Input Agriculture podcast is a solo passion project built with my ample time and meagre resources. If you would like to support the show, then sign up for a paid subscription to my Zero Input Agriculture blog at Substack. As a reward, you can read all my past posts and listen to the audiobook of Taming the Apocalypse. If you prefer reading about the past and future of biological technology, you can read the ebook of Taming the Apocalypse through Amazon. And finally, please rate and review the show on whatever podcast app you use, since it helps other people find me. Or recommend the show directly to anyone who could use some inspiration to become a plant breeder themselves.